Hello, everyone, and welcome to today's Flash Talk. I'm your host, Dennis Koester. I'm the Director of Industry Research and Relations here at Drug Hunter, and I'm here with our guest, Yoshi Ishihara, today. Welcome to the show, Yoshi. Pleasure to have you today.
Hi, yeah.
I will introduce our guest, Yoshi, now. Actually, his name is Yoshihiro Ishihara, but, short Yoshi. And Yoshi obtained his bachelor's and his master's degree from McGill University in Canada and received his PhD under the supervision of Phil Barron at Scripps in La Jolla, California. And he was working on natural product synthesis and methodologies involving heteroaromatic compounds. Yoshi began his career as a medicinal chemist at Vertex Pharmaceuticals, where he focused on hit-to-lead and lead optimization for ion channel programs, including projects on cystic fibrosis, as well as on the now famous NaV1.8 compounds for acute pain. And he later worked at Vividion Therapeutics, where he specialized in chemoproteomics, library design, and medicinal chemistry with a particular emphasis on covalent chemistry. Today, he's at Genesis Therapeutics, and there he leverages the power of artificial intelligence for drug discovery. And Yoshi continues to study the role of small substituents in drug discovery to deepen his understanding of medicinal chemistry, and we will hear more about those, the chlorine and the methoxy group today. So it's an honor for us to have you today. Welcome, Yoshi. It's great to have you.
Thank you. Yeah. I honor the invitation as well. Thank you so much. I'm happy to be here.
Awesome. Yeah, and as you know, like as the warm-up, we typically ask a few questions to warm up. So I was wondering, Yoshi, what inspired you to start your career in drug discovery?
Yeah, so I had to think about this actually, because at the beginning, like I just liked organic chemistry, really. I was really into NMRs actually, and that got me into organic chemistry in the first place. But because of NMRs, I like small molecules and conformations, right? Conformations can affect an NMR spectrum a lot, and so then I got into how small structural changes can make such a big difference in a protein binding pocket. And so I got into drug discovery more from like a ligand protein interaction point of view.
Oh yeah, that's probably visible today in the presentation, right? Yeah, I
Yeah,
love
yeah, I
that.
still like it today. Yeah.
Yeah, I love that. That's amazing. And what's your favorite molecule? Is it like maybe a favorite molecule because of a specific interaction or just
No, for me it was like stereochemical complexity, weird, arrangement of heteroatoms and stuff. So I used to be really into complex, like bioactive natural products, right? That's what I
wow.
did in grad school. So like Taxol or calicheamycin or helicondrin B, right? That's the natural product basis for Halavan. But I
Yeah.
still like complex structures. So it doesn't have to be a natural product. It could be like Merck had this like macrocyclic peptide like MK0616, right? So that's
Correct.
quite
Yeah.
the beast of a molecule, which it must be hard to make, but it's nice to see. But I also like smaller structures with like unique functional groups. I think Gilles Ouvry was mentioning this too, but asciminib, right, that has an OCF2Cl group. It's pretty unique. Um, and then finally, of course, I like the Vertex cystic fibrosis drugs in the triple combo therapy, right? I mean, I do have a better understanding, I guess, of them, and I like them structurally as well.
Yeah, no, we talked about a few of them before. There was there quite some cool structural motives in the coming one. So super cool. What are you most excited about in drug discovery today, Yoshi?
So I'm still kind of a traditionalist when it comes to drug discovery. So I still like small molecule drugs the most. But I do find it impressive, right? How scientists can come up with like so many creative ways to tackle drug discovery. So I'm talking about like different modalities that exist. So whether it's ADCs or bifunctional degraders or booze or radiopharmaceuticals, even AI in drug discovery, right? So that definitely excites me in drug discovery, especially today.
Yeah, no, a lot of really exciting things on the cusp of breaking through today, right?
Yeah.
Yeah, sounds great. So if you could give yourself some career advice, your younger self, what would you tell your younger self to do more of
Yeah,
or
so
less of,
it's
maybe?
well, not really less of. I mean, I was always inclined for science. I wasn't good at much else. I think chemistry, science and chemistry, resonated with me kind of early on as well. And from grad school, I don't regret focusing as hard as I did in organic chemistry during grad school and postdoc. But for me, the first few years at Vertex, I feel like I didn't do enough sort of broad reading, if you will. As a med chemist, right? Like you don't need to be an experimental expert in everything, but you need to be conversational on different topics. And I think I started late on that sort of broad reading in other fields. So if I had Drug Hunter back in 2015 when I started my industry job, I think that would have helped me a lot.
That's amazing. Yeah, thank you so much, Yoshi. And what do you like most about Drug Hunter?
Yeah, and this too, like, so you always ask your panelists this question, and I like so many things about Drug Hunter, right? I'm quite the fan. I like looking at the effect of small substituents in drugs. So this one's the same answer as Gilles. So I like the substructure search, of course.
Yeah.
I love the molecules of the month. It's just a nice collection, right? Or molecules of the year. But maybe the most important one or useful one to me is the first disclosures from the conferences, right? Because I would, of course, like to go to all the conferences, but no one has the time for this. So Drug Hunter is making sure I don't miss out. And ultimately, like Drug Hunter is about saving me time, right? I'm always someone who says like, oh, I don't have time for this. Like I don't have time to read everything I want to learn about. And so Drug Hunter allows me to learn a variety of topics in a short amount of time. So I listed a couple of things. Maybe the time component is the most important one for me in the end.
Yeah, that's awesome. Yeah, yeah, we definitely do like to put those conference reports together. But yeah, I did want to before Yoshi starts his presentation, I did want to show you a relatively new feature of Drug Hunter that also I think would save people a lot of time. So if you navigate here to resources and then go to patent search, we actually also put together a list of patents that you can search. So for example, if you're working in the field of oncology, just go here and type oncology, and you can really see just by scrolling through here some of the targets that people are currently working on in the field of oncology. And without opening the patent, you can directly see what are some things that are notable from the patent. So how many compounds are in there. Here, for example, is a compound with 74% tumor growth inhibition in a xenograft model. So you can directly see whether or not this is a relevant patent, and you can get some inspiration for new targets. Or if you're following a target and that can be really beyond the targets you are working on yourself, you can just type this target up here and see what is going on in the field. Again, you can, on the right-hand side, quickly see an overview of what is in those patents, what is the disease area, and who is working on the particular field. So just give this a try, after our webinar here, and let us know how this works for you. And with that, I will hand over the keys to Yoshi for his super interesting talk on the roles of the chloro and the methoxy group in drug discovery. That will be a very interesting talk. Yoshi, the stage is
Great.
all yours.
Awesome. Yes. Oh, yeah. And good point about the patents. I haven't used that feature too much myself, but I'll certainly look into it. That's definitely a time saver. So thanks for sharing that with us, Dennis. Well, so yeah, thanks, Dennis, for that very kind warm-up conversation. I really like this Drug Hunter community. So I'm very happy to be able to discuss my topic with this audience of scientists from around the world. Today's talk as well, the title of the talk as well as the concept revolves around two small substituents, chloro and methoxy. This presentation is based on a JMedChem paper as well as a European JMedChem paper that I wrote recently with my co-author Deborah, and I would like to share with you the takeaway messages of these papers. I like the Drug Hunter flash talks because I feel that they're a bit more personable than a traditional seminar. And so I'd like to start by sharing with you what I find to be inspiring in the drug discovery world. This first one is a classic. It's a paper called A Medicinal Chemist's Guide to Molecular Interactions. It's a JMedChem in 2010, written by Stahl and coworkers from Roche. This paper covers all the different interactions you can imagine between a small molecule ligand and a protein binding pocket. So if you're a medicinal chemist, this paper is a must read. Here's some other journal articles that I find inspiring. This first one is what I consider to be perhaps more recognizable when it comes to the concept of magic methyl. This paper was written by Schoenherr and Cernak in 2013. I was still a postdoc and I didn't know much medchem at the time, but I remember being impressed at how such a small substituent can result in such a dramatic potency effect. So then I got interested in reading more about small substituents in drugs, and 2015 was about the time that I had joined Vertex for my first industry job. Of course, Nick Meanwell is a very prolific writer, and it's hard for me to pick one of his favorite one favorite Meanwell paper, but these two papers really cemented in me the understanding of the fluorine atom in drug discovery. And this third paper, this is again a small structural change that leads to a large improvement in potency or ADME. So here's our very nice sort of catchy title, right? You have magic methyl on the left and you have necessary nitrogen on the right. This paper was written by Lou Pennington, who is currently with Drug Hunter. As for other sources of inspiration, I go to Daryl McConnell's MedChem page. He writes about a variety of topics, but for example, he has something called the strength of hydrogen bond interactions. And he does so in a very visually pleasing manner. He's also an artist, so he uses these hand-drawn images to illustrate concepts in drug discovery. Next up, we have Gilles Ouvry. Last week, we heard from Gilles in an excellent Drug Hunter flash doc. What are equally excellent are his LinkedIn posts. He's a guru when it comes to molecular torsions and conformations, and he puts these together in concise but very informative posts. So I truly, truly recommend following Gilles on LinkedIn. And of course, when it comes to online inspirations, we have Drug Hunter. I know we already talked about a couple of reasons I like Drug Hunter, Dennis, but it turns out I forgot one. This is, and I want to highlight this here. There's a lot of nice free resources on Drug Hunter as well, for those of you who are not subscribed. And that could be DMPK cheat sheets, or that could be summaries such as this Topliss Tree. So Topliss Tree, for those of you who are unfamiliar, is a visual guide on how to explore SAR around a benzene ring. So when you have an unsubstituted phenyl ring, the Topliss Tree recommends to first make the para-chlorophenyl. If this chloral compound is more potent than the parent hydrogen molecule, so the unsubstituted phenyl, then make the dichlorophenyl. If the chloral compound is equally potent as the hydrogen, make the paramethyl. And if the chloral compound is less potent than the hydrogen, then install the methoxy group. Now, this Topliss Tree was established in the 1970s, and there are more recent revisitations and notes, but this original Topliss Tree is still being used in medchem groups around the world. But there are some caveats. For example, the Topliss Tree recommends you to make and assay molecules one by one. But in modern drug discovery, we really don't have the time to just make one molecule per design cycle. So it makes more sense to make a batch of molecules together, right? And to be more efficient in your synthesis. And so that leads us to the batch-wise approach of the Topliss Tree. Topliss Tree, or Topliss says, instead of just making the para-chlorophenyl, make these 5 compounds at the same time to take more informed decisions through batched synthesis. Now, I'm not going to go through all these QSAR parameters that go into this approach. The QSAR parameters are shown here. But what I want to show in this slide is that the most important substituents are methyl, chloro, and methoxy. A lot has already been written about the methyl in the form of magic methyl, for example. So I wanted to focus, and I got quite fixated on the substituents of chloro and methoxy, and I wanted to learn more. But it turns out that there's not too much in the literature on chloro and methoxy in drug discovery. There's only one review article for chloro and one perspective article on methoxy in the context of phenol ethers. Now, these 2 articles were useful in that they showed lists of approved drugs containing these substituents, but they did not go too deep in the reason behind these substituents being included in these drug molecules. So here's a quote from someone I looked up to. It says, if you're looking for information on a topic, but you can't find it easily, then you should write your own review on it. Now, this is not a quote, it's a paraphrase, but I'm sure Nick Meanwell would concur. And so we wrote our own papers on this. On the left, we examined matched molecular pairs in a paper that we call Magic Chloro. On the right, we studied the role of the methoxy group in methoxy-containing approved drugs. Now, overall, I consider chloro to be magic, but methoxy not as much, and I'll get into that a little bit in this presentation. Okay, so today I'm not trying to advocate that chloro and methoxy are these rare substituents, and that after this presentation today, there'll be more drugs containing these groups. I'm not trying to say that. Chloro and methoxy are already quite prevalent in approved drugs. When you look at Neodarson's top 200 posters list, even amongst the first 30 drugs or so, you'll find examples with chloro, methoxy, or even both. But what is most important to me, and the question I had, is why were these small substituents included or installed in these drugs? Now, the answers could be many. It could be potency reasons, like ligand-protein interactions. It could be metabolic improvements for the case of chloro, and physicochemical properties in the case of methoxy. A really good place to learn about ligand-protein interactions is the protein data bank. So I include the PDB numbers here for these drugs, and I'll talk about some of these X-ray co-crystal structures in the upcoming slides. Well, let's first talk about electronics a little bit. When you look at electrostatic potential maps, usually in blue are indicated areas that are electron poor or delta plus charge. And in red are indicated areas that are electron rich or delta minus charge. For chloro, I will compare methane to chloromethane. Methane doesn't have much charge separation in its molecule, so you don't really see strong colors. But for chloromethane, right, chlorine is electron withdrawing, so it has a strong red color here around the chlorine atom, like a band. This is called the negative belt. And you'll realize that this strong red color doesn't propagate all the way through to the tip of the chlorine atom. This head here is actually not electron rich, but it's rather slightly electron poor. It's slightly delta positively charged, and it's called the sigma hole. On the methoxy side, I'll be comparing benzene to methoxybenzene. Now, benzene is a nonpolar molecule, but it is electron rich at the center, above and below the ring, and then it is slightly electron poor at the periphery. Now, when you look at methoxybenzene, you'll see that the red electron density propagates through to the oxygen atom. And as a counterbalance, I guess, you see this blue area of electron poor regions close to the methyl group. Now, this is not quite a sigma hole, but it is a delta plus region of the molecule. So to summarize these effects, chloro can present a delta negative charge through its negative belt and a delta positive charge through its sigma hole. Whenever chlorine's on a benzene ring, it makes the aromatic ring electron poor, so it can engage in interactions as an electron poor aromatic ring. And of course, chlorine is quite the lipophile, so it can have lipophilic interactions as well. On the methoxy side, it presents a delta minus charge through the oxygen atom and a delta plus charge through the terminal methyl group. Again here, in an opposite fashion, when you have a methoxy group on a benzene ring, it makes it electron rich, and so it can interact as an electron rich aromatic ring. So next we have the question of how do these aspects of chloro and methoxy engage with the protein binding pocket? So when you have a delta negative charge on a chlorine, it'll interact with the opposite charge, so a delta plus charge on a protein. That delta plus charge on a protein could actually come from an aromatic ring as well. So it's the quadruple moment of an aromatic ring. An aromatic ring has a delta minus charge in the form of pi clouds above and below the ring, and delta plus charge surrounding the ring in plane with the aromatic ring. So now I'll fill this table in matrix fashion, and you have all these molecular interactions. I know it's a really busy slide, so I'll go through these one by one. So this first column, chloro presents a delta negative charge, interacts with a delta plus charge on a protein, and that could be in the form of a hydrogen bond donor. If that's the case, this is called a hydrogen bond. The hydrogen bond length is typically between 2.8 and 3.5 angstroms, and I'm counting the heavy atom to heavy atom distance here, so chlorine to oxygen. I'm disregarding the hydrogen for these bond lengths. A hydrogen bond strength is typically between 1 and 10 kcal per mole, and for chlorine, it's less efficient as a hydrogen bond acceptor, and so it's usually at the lower range or lower part of this range. For reference, 1.4 kcal per mole is about a 10-fold improvement in potency. You can see in this X-ray co-crystal structure of the anti-cancer drug vismodegib, 2 hydrogen bonds at about 3 angstroms each, and they're both trying to avoid the sigma hole. They're trying to engage both with the negative belt of chlorine. Next up, we have the orthogonal multipolar interactions. These are interactions between the negative belt of chlorine with the electropositive area of a carbonyl group. So that would be the carbon atom. This bond length is a little bit longer, and it's weaker than a hydrogen bond. You can see that in this drug tafamidis. The chlorine atom is engaging 2 carbonyl groups at the electropositive carbon atoms. This tafamidis was one of the chlorine-containing drugs in that Neodarson poster slide. That delta negative charge can engage in weak CH to chloride interactions. It engages the CH bond of an arene, the CH at the edge of the arene. Now, there's a geometric requirement here in that the chlorine atom needs to be coplanar with this aromatic ring. And you see that in this antipsychotic drug, cariprazine. The X-ray structure shows chlorine in line or in plane with this phenylalanine. In this next column, we have a chloro presenting a delta plus charge through its sigma hole, and that engages a delta negative charge, the opposite charge in a protein. That could be often through the oxygen atom of an amide backbone carbonyl. So this would be called a halogen bond. Its bond length is typically that of a hydrogen bond, so about the same, but the bond strength is a little bit weaker. The geometric requirement here is that the carbon, chlorine, and oxygen atoms need to be collinear, and you can see that in the X-ray structure of the erectile dysfunction drug avanafil. The carbon, chlorine, and oxygen atoms are collinear. I think the bond angle here, I think I measured it at one point, I think it's like 170 degrees or so. If you get lucky, you can also get this dual hydrogen-halogen bond. So in this X-ray structure, you can see that the carbon, chlorine, and oxygen atoms are nicely aligned. And the chlorine atom is using its negative band to interact with this NH in a hydrogen bond as well. The delta plus charge can engage with the pi cloud of an aromatic ring. This is called a halogen pi interaction. And the geometric requirement is that the CCl bond has to come in perpendicularly or orthogonally to the plane of the aromatic ring. You can see that nicely in this X-ray co-crystal structure here. So chlorine makes an aromatic ring electron poor. Now, the strongest pi-pi interactions arise when you have an electron-poor aromatic ring stacked on top of an electron-rich aromatic ring. And that can come in sandwich form or parallel displaced fashion. In the X-ray co-crystal structure on the right here, we have the electron-poor dichlorobenzene ring sandwich stacked on top of the electron-rich pyrrole portion. And it's offset, it's parallel displaced with the benzene portion of tryptophan. This top benzene ring, you could actually rotate that and make it actual, and you can get a T-type interaction as well. If you rotate this a little bit, you get an edge-to-face interaction. A paper by Kim in JACS 2005 says that the systems with electron-accepting substituents, including chloro, favor the actual conformation. So if you look at this X-ray structure, the electron-poor ring sits actually, while those with electron-donating substituents favor the facial conformation. So usually the electron-rich ring sits at the bottom and shows the face of the ring to the ring above. Chlorine is quite the lipophile, so it engages in numerous van der Waals and hydrophobic interactions. It's quite efficient at that. What is quite convenient about these interactions is that you can get the design wrong. You can design it like the left and actually get the right, and this will still give you favorable interaction energy. What is also convenient as well is that these interactions operate at longer distances, but individually they are weaker. So a lot of these interactions have to add up to give you a significant potency effect. In the X-ray structure of this anti-cancer drug, lenvatinib, you can see how the chlorine atom is nestled nicely in a lipophilic pocket formed by the valines and leucine, and it's forming multiple van der Waals interactions. Okay, now let's move on to the methoxy side. It's a similar story here, similar types of charges and interactions. In this first column, methoxy presents a delta minus charge, so the oxygen atom forms a hydrogen bond. And you can see that in the X-ray structure of this anti-psoriasis drug, apremilast. It can also engage in orthogonal multipolar interactions. So the delta minus charge in the oxygen interacts with the carbon atom of a carbonyl group in this antihypertensive drug, aliskiren, binding to its pocket. The delta minus charge on the oxygen can engage with the CH at the edge of an arene. So in this pain-relieving drug, naproxen, the oxygen atom is in plane with the tryptophan of, this is COX-2, I believe, the protein. In the second column, methoxy can present a delta plus charge through its terminal methyl group. It can engage the oxygen atom of an amide backbone carbonyl, and this can lead to a CH hydrogen bond. You can see that in this anti-cancer drug, osimertinib, the methyl group here is engaging the carbonyl oxygen atom. But that delta minus charge doesn't have to be an oxygen. It could be a sulfur atom as well. So in this X-ray structure, you see that the methyl group is engaging with the cysteine sulfur atom. That terminal methyl group with an electron positive charge can engage with the pi cloud of an electron-rich ring. This would be called the weak CH to pi interaction. You see the geometric requirement of OC bond being axial or orthogonal to the aromatic ring, and you see that in the X-ray structure of this anti-cancer drug, elacestrant. The methoxy group can make a benzene electron rich, and so that can again engage in a sandwich type interaction or a parallel displaced interaction. And you can see actually both of them in the X-ray co-crystal structure of the anti-insomnia drug daridorexant. The methoxybenzene interacts in a sandwich fashion intramolecularly and in a parallel displaced fashion intermolecularly with this histidine imidazole ring. You can flip that top ring over and you can have a T-type interaction or an edge to face interaction. Again, based on Kim's Jack's paper, the more electron-rich ring usually sits at the bottom, showing the face of its aromatic ring to the ring above. So this is the edge-to-face interaction. In this X-ray of papaverine, you actually see a second pi-pi interaction as well, and this would be a parallel displaced interaction. And finally, a methoxy group overall is not very lipophilic. The delta log D of a methoxy group is essentially net zero. So it's not very lipophilic, but the terminal methyl group has some lipophile kind of element, so it can engage in some van der Waals interactions, but they're not that strong, so I do indicate them in a lighter gray font here. So I'm showing all the interactions here again in this overwhelming looking slide. But my take-home message from this slide is that chloro and methoxy really have a rich variety in the types of molecular interactions they can engage in. And this contrasts well with the methyl group. The methyl group is considered magic when it comes to potency, but it does not benefit from all these different types of interactions. Okay, moving on. So I'll show some examples of significant potency improvements when you have chloro or methoxy in your lead molecule. In the left example, when you have chlorine at this position, the authors get a 1,300-fold improvement in potency against this HIV mutant and a 179-fold improvement against another HIV mutant. In this situation, the authors know exactly what is behind this potency improvement because they obtained an X-ray co-crystal structure. First, you can see that the chlorine atom is engaging in an intramolecular hydrogen bond with this NH. So that locks this bond over here and presents the carbonyl oxygen parallel with the imidazole NH. So you can see that parallel interaction and this parallel hydrogen bonding to lysine 103. Chlorine also makes this imidazole ring electron poor, so it enhances the acidity, so lowers the pKa of that NH, and that enhances the hydrogen bonding interaction as well. Furthermore, the chlorine atom can engage in a halogen bond with the leucine carbonyl and an orthogonal multipolar interaction with the carbon atom of this carbonyl. And finally, this chlorine atom is nestled nicely in a lipophilic pocket formed by phenylalanine and proline residues, and so it engages in multiple van der Waals interactions. So overall, looking at these numbers, I would consider this a significant chloro effect. Now, this example is quite interesting, one of the more interesting ones in the paper, where this molecule has a ferrocene in it and has very significant numbers. The biggest numbers of potency improvement that we've seen in the paper is from this reference. And so in this example, putting in a chlorine atom at this position led to a 100,000-fold improvement against this cancer cell line and a 10,000-fold improvement against this other cancer cell line. And the author was quite just nonchalant about it. He was like, we installed this chlorine atom and we obtained a potency improvement. And sort of left it at that. I think there's much more to it for such a dramatic potency improvement. There must be a big conformational change due to the inclusion of or installation of this chlorine atom. So first, while the chlorine atom is at the ortho position to a biaryl, so I'm sure it's changing this biaryl angle, right? So changing that conformation. But there must be more to it. There must be some, overall change in conformation or something for this molecule. So maybe this chlorine atom is starting to, engage with this metal center and changing the overall conformation. I don't really know. But what I do know is that, well, when you have such a dramatic effect and such an unexplainable potency improvement, for me, that signifies a magic effect. On the methoxy side, this is SAR exploration en route to the antiviral drug Fostemsavir. The authors, when they included a methoxy group at the C4 position, observed a 294-fold improvement in potency. Other substituents, such as fluoro and chloro, led to improvements in potency, but not as much as the methoxy. And putting in something larger like isopropoxy totally killed the activity. Looking at this X-ray co-crystal structure, you can see that the methyl group is engaging in multiple van der Waals interactions. But I think the bigger point about the substituent at this position, it changes the conformation of the right-hand side of this molecule. Maybe that binds it better to the protein pocket. So this number, 294-fold, I think it's a significant methoxy effect, but perhaps not magic. And finally, we have this example. I think this is a CNS program, if I remember right, where installation of a methoxy group at this position led to a 28-fold improvement in biochemical potency. Surprisingly, this led to even better improvement in cellular potency. So they obtained a 75-fold improvement in cellular potency. So this discrepancy, if you will, comes from the permeability. So when you look at passive permeability, the methoxy improves the passive permeability of 5-fold and also reduces the efflux ratio by 4-fold. So these are all both positive improvements. So because the methoxy compound is more permeable, it permeates the cell membrane better and it performs better in a cellular assay. Structurally speaking, the methoxy group engages in an intramolecular hydrogen bond with this NH, so it effectively masks this hydrogen bond donor, and that's why you get a better permeability. I think the authors actually obtained an X-ray co-crystal structure of this molecule, and they see that intramolecular hydrogen bond, and that hydrogen bond was part of their strategy in their lead op campaign. So to wrap up the potency section here, chloro can lead to significant or even magic potency gains akin to a methyl group. Methoxy can sometimes lead to potency improvements as well. All in all, I think chloro and methoxy are arguably the best scouts for pocket finding, and that's because of the various types of molecular interactions they can engage in. But for chloro, do watch out for the lipophilicity. So you might want to consider calculating LLE or LIPE, right? The ligand, lipophilic ligand efficiency instead of just potency. Okay, so moving on to metabolism, overall chloro is superior to methoxy. In this example on the left, installation of a chlorine atom at this position led to a 20-fold improvement in vitro half-life and clearance in microsomes. For fluorine, that also led to good ADME, but it had a loss in potency. In contrast, methyl group here was better for potency, but was worse for ADME. And so chloro was really the perfect balance between the ADME effects of fluorine and the potency effects of methyl. Now, on the methoxy side, methoxy is often frowned upon for metabolic liability, and it's warranted, right? When you have a methoxy group on a benzene, it often gets O-demethylated with phase 1 metabolism, leading to a phenol, and that can get glucuronidated and cleared rapidly from the body. But there are some ways to address this issue. Maybe you can lower methoxy metabolism by playing with electronics or sterics. On the electronic side, when you have a methoxy group on an electron poor ring like pyridine, this has a slower rate of O-demethylation compared to a methoxybenzene. I personally haven't seen examples of a methoxy on a pyrimidine being O-demethylated, so I think the electron poor nature of this ring helps with mitigating O-demethylation. Now, if the electronics don't work or it's not tolerated for potency reasons, you can introduce sterics, right? You can have a bulky group at the C2 position or block both the C2 and C6 position on the benzene ring, so just sterically hindering out the O-demethylation process. Now, if both of these options don't work out, maybe you can install methoxy isosteres, right? Maybe you can find a pocket using a methoxy group and then change it after to mitigate metabolism. So you could replace the entire methoxy group with a fluorine, or maybe just the oxygen atom with a CF2. The CH bonds of the methoxy is what is oxidized. So maybe you can replace the CH bonds with a C deuterium bond or a C halogen bond. Now let's spend one slide on physicochemical properties. Also, off-target potency like hERG is closely related to physicochemical properties, so I'll cover that here as well. And here I'd say, methoxy is superior to chloro, okay? All the data on this slide is from an excellent paper for matched molecular pair comparisons from Gillett and coworkers. There's also a paper referenced below by Dean Brown as well, so make sure to check those out. They're very informative. Now, when you look at chloro, right, when you look at the individual parameters of lipophilicity, solubility, and hERG inhibition, when you go from hydrogen to chloro, it doesn't lead to a favorable outcome very often. All right, versus most of the time, it leads to an unfavorable outcome. So this is a big disadvantage for the chloro. If you can replace chloro or swap chloro for a fluoro and still get the same potency, I would definitely choose the fluoro, okay? And looking at physical chemical properties, so these parameters here, it's still unfavorable for fluoro, but not nearly as badly as for chloro. Now, on the methoxy side, I think methoxy is quite neutral. By neutral, I mean it doesn't really positively or negatively affect these parameters. So that's actually a good thing. So if you can install a methoxy group onto your molecule, right, and get a potency improvement, but not suffer in terms of metabolism, then by all means, you should install that methoxy group on your molecule. But if you want better properties, you might want to choose hydroxy. Right? When you have hydroxy, everything is favorable across the board in terms of physical chemical properties and hERG. The only downside or major downside is that hydroxy often makes the potency worse when you have matched molecular pair comparisons on average. And it makes your metabolism pretty bad sometimes, right? You can get glucuronidated and cleared. So in general, I do recommend methoxy over hydroxy for these reasons. There are unique advantages of chloro and methoxy in drug discovery. I was talking about how chloro can be a scout to find pockets inside a protein. And halogen-containing fragment libraries are becoming more popular these days. So HEFLibs, that's a halogen-enhanced fragment library, Fraglites, and the Halo library becoming more popular. And so for me, a fragment is like a very small molecule, right? It has a very small molecular footprint, so it doesn't have much binding affinity to the protein pocket. So bigger atoms like chlorine could help enhance that potency. And this is also what's unique about chlorine. Halogens heavier than fluorine can facilitate crystallography, right? You can see more electron density around that atom, so it's easier to look for in an X-ray crystallography. And so you can grow co-crystals of your fragment inside the protein binding pocket. If you can combine fragments together, for example, you can do some fragment growing exercises, so it really helps the fragment-based drug discovery process. On the methoxy side, I'll talk a little bit about PSA. So that's polar surface area. For me, PSA is like a finite amount, right, of polarity currency, if you will, that you can use. And that's about 140 angstroms squared. And you can use that onto your lead molecule until you run out of your chances of developing an orally bioavailable drug. So methoxy is nice because it's a hydrogen bond acceptor that can do a variety of things, but has a very small PSA footprint of 9 angstroms squared. In contrast, hydroxy or carbonyl or methyl sulfone even, these have a much higher PSA. The idea of PSA reduction was used for deucravacitinib, which is a recently approved drug against psoriasis. So the BMS scientists here had this sulfone-containing sub-nanomolar compound. But they wanted to explore this pocket further. They wanted to grow into it. But then they realized this methyl sulfone is costing them a lot of PSA. So they decided to swap this sulfone out for a methoxy group. The role of this sulfone was to displace a high-energy water molecule from the binding pocket. And so methoxy can do the same thing. However, they observed the 5-fold loss in potency. But at the same time, they were able to recover or gain back 25 angstroms squared of PSA. So they continued on with this series of molecules. Then they explored this northern pocket, grew out into here, and they were able to get molecules with better potency. You can see the methoxy effect in this matched molecular pair, right? It has a good, 60 or 70-fold improvement in potency thanks to the methoxy group. And then this finally led to deucravacitinib, and they were able to keep that PSA under the typically recognized value of about 140 angstroms squared. That's kind of like the range you want to, or the maximum value you want to keep. You want to stay under that in order to make your compound orally bioavailable. So with that, I'll come to the conclusion side. Chloro and methoxy are great scouts for potency improvement. I think chloro is like a mix of methyl and fluoro. So it has the magic potency effects as well as the metabolic blocking effects of fluoro. I say include chlorine in molecules if the potency improvement is 10-fold or better per chlorine atom, and certainly if it's 30-fold or better. And I include that 10-fold minimum because you want to account for the lipophilicity of chlorine. You should also include chlorine when fluorine can include PK or ADME, doesn't have great potency. And you might want to try ortho and meta substituents, not just para, because there's nothing inherently special about the para position. Dean Brown says it's just because we have a love affair with the parachlorophenyl. So I'm going to try to speed up a little bit here because we're running out of time. There are some caveats to the chloro, but the biggest caveat is that aqueous solubility becomes very poor when you have a chlorine atom or multiple chlorine atoms. So if it's too poor, you might want to include fluorine as a better option. And overall, I would keep the total number of chlorine atoms to 3 or less. On the methoxy side, methoxy is advantageous because it has a 0 delta log D and a small PSA footprint. Like the chloro, I try orthometa substituents, not just para, and I'll keep the total number of methoxy groups to 3 or less. And the main really caveat for including a methoxy group is that metabolic instability, right? So OD methylation, and I showed some strategies on how to mitigate that OD methylation. So this acknowledgement slide, there's a lot of list of names here. These are the people who really shaped my chemical upbringing, right? They were the people who kept making me ask those fundamental questions, right, at the molecular level. So from Scripps, I have my mentor, Phil Barron, and many others. From Vertex, I have the late Peter Grutenhuis and others. From Vividian, Benji Bryan, and many more. And at Genesis, there's Martin, there's Gianpaolo, Brett, Indra, Shifang, Adi, Yongkai, Chris, and many others that I get to learn from every single day. When I was putting together this list of names, I realized that a lot of these are computational chemists. And so my hat's off to the computational chemists in the crowd. You really have an expert understanding of molecular interactions. Additional thanks to Gilles Ouvry for sharing your knowledge through LinkedIn posts and through messages as well. And I don't know how I had the pleasure of meeting Nick Meanwell and being mentored by him, if you will, for paper writing. So I thank him for sharing his experience and for his dedication to the MedChem community education. And then finally, thank you, Drug Hunter, for your invitation for me to present today. And for those of you in the audience, thank you all for your kind attention.
All right, Yoshi, thank you so much. It was a fantastic overview of the molecular interactions and, you know, the physicochemical parameters of chloro and methoxy groups. Really fascinating. And one of the things that I just noticed on your slides is that osimertinib that you highlighted with the methoxy group was actually just approved yesterday. I mean, it has been approved earlier, of course. But yesterday, the FDA issued an additional approval in very specific indications for unresectable stage 3 non-small cell lung cancer. So really
nice.
super interesting. Yeah, so we do have a lot of questions in the audience already, so let's go through them. For example, Simon, he's wondering if it's possible for chloro to engage the target in a covalent inhibition mechanism, and how would such a chloro-containing warhead look like?
Yeah, so we talked about this a lot. And actually, my apologies for this presentation. I was trying to put together chloromethoxy and do it in a balanced way. And that made me talk about aromatic chloro and methoxy, but I didn't really talk about aliphatic chloro and methoxy, right? And so when you have, especially for an aliphatic chloro, so when you have primary alkyl chlorides, especially when you have a nitrogen, carbon, carbon, chlorine, right? This is a typical covalent warhead. So you can get a covalent warhead that way. Not that all aliphatic chlorides are bad, right? As Gilles talked about this a little bit, tertiary alkyl chlorides are perfectly stable. They're great. Even secondary, a lot of them can be stable, but you know, caution there. Primary alkyl halides, you do have to watch out for. The more known covalent warhead for chloro would be alpha to a carbonyl group. Those are hot warheads, often used in probes, for example, when you have an alpha chloroacrylamide, alpha chloroacetamide, it is quite commonly used as a covalent warhead.
Yeah, and probably also chloropyrimidines could maybe sometimes be covalent, right?
Yeah, you can have an SNAR mechanism as well. So if you have, yes, a very electron poor ring, you can get that. You know, sometimes people are worried about having a chloro at the 2 position of a pyridine as well. I think that's context dependent. But chloro is not as good of a leaving group as fluoro, for example, on an aromatic ring. So I'm not as concerned for chloro than for fluoro for SNAR mechanisms.
Awesome. Yeah, cool. Thank you so much. Then Laura is wondering, can you further elaborate on why a reduction in clearance is observed when a hydrogen is replaced by a chloro, even if lipophilicity is increased?
Okay, so let's think about that. So when you have, let's say, methoxy, right, and you're changing the CH bonds to, let's say, a CF to Cl or something like, people don't usually put OCCl3. That's usually uncommon. That would be quite lipophilic. And yes, I was mentioning earlier too, like lipophilicity, of course, affects many other parameters too. That I think will have to be a case by case. You'll have to see how much lipophilicity is being put into your molecule. I don't remember about the CF2 chloride, but OCF3, for example, is much more lipophilic than OCH3, right? But like a log unit of 1, log D of
Hmm.
1. So yes, lipophilicity can engage, like let's say CYP inhibitions as well, and you can have other issues. So um, I'll have to think about an, specific example where blocking CH bonds but led to worse metabolism. You might be directing your metabolism to other areas of the molecule, not necessarily the methoxy group at that point, but because of its lipophilicity, it binds to CYPs better and then maybe gets metabolized elsewhere in the molecule.
All right. Yeah, let's see about the next question. So how do we evaluate the methoxy groups attached to an aliphatic carbon where the lone pair is not conjugated in terms of ATME and physicochemical properties?
Yeah, I have. So this is not exactly quite what you're talking about in this, but so methoxy on an aliphatic carbon, right? And I was planning to show this, and I didn't really have time. It
Mhm.
has slightly different, bond angles, right? And
Nice.
methoxy on a benzene, right? There's only one kind of available lone pair. In aliphatics, you have two available lone pairs. Um, Typically, from what I've found, methoxys that are aliphatic are less oxidizable than on a benzene ring. So if you just
Mhm.
compare straight up like methoxycyclohexane versus methoxybenzene, like the methyl on a benzene could get demethylated faster. But it depends on how sterically hindered this is. If this is on a primary alkyl group, right, that's very exposed, and so you can get metabolism at the methoxy group.
Awesome. Yeah, thank you. And I guess we have a follow-up question on the sp3
Ah,
chlorobond. So Anne Tran says, thanks for a great talk, Yoshi. You mentioned examples of chlorophenol, but I'm curious your thoughts on CsP3 chloro, which is not common in my experience. In contrast, alkyl fluoro is really common and very beneficial in my experience.
and
So
absolutely, I agree. Again, this goes back to me not talking about aliphatic chlorines. Maybe that could be a separate kind of small talk, um, or a shorter talk. Yes, so tertiary alkyl chlorides, especially the one that Gilles mentioned with the chlorocyclopropane, those are perfectly stable.
yeah.
Tertiary alkyl chlorides are great. I think they're good isosteres of a tert-butyl, if you will. Secondary alkyl chlorides, like I said, aren't too bad, and people have a tendency to feel that an alkyl chloride is very unstable. But when you think of molecules like even dichloromethane, like it's CH2Cl2, and it's fairly stable as a molecule, all in all, like considering like sure there's toxicity and all that, but or sucralose, right? It has a lot of primary and secondary alkyl chlorides, but it's still quite a stable molecule and it gets excreted unchanged from your body. So I'm not too concerned about CSP chlorides, especially CSP3 chlorides, especially if it's on a tertiary system and even on a secondary system.
Yeah, makes a lot of sense. I have a question on my own, actually. I was wondering, in terms of the molecular interactions, you beautifully illustrated those with the chloro as well as the methoxy groups. I was wondering, do you think that the fluoro interactions, would they be similar to the chloro type interactions?
So they are for the delta minus charge, but it doesn't have that sigma hole, right? So that's the main
Hmm.
difference between
Interesting.
fluoro and chloro. I think bromine is more similar to chlorine in that regard. But I have an overall kind of cutoff, right? I think like maybe 5 fluorines should be maximum in a molecule, 3 chlorines should be maximum in a molecule, and bromine, like you want to keep it to 1 at most or less. You don't really want to put bromines on a molecule, so you can maybe get a magic bromo effect, but it's not really like a recommended truly group to have in there.
Makes sense. I did see molecules that have upwards of 10 approved fluorines that are
Uh,
actually there. So, for example, look at lenacapavir. uh,
yeah.
Another question from Sebastian Young is concerning the metabolism of the methoxy compounds.
Okay.
What about the possibility to form reactive quinide structures after demethylation? I was
I
actually
mean,
wondering that too.
absolutely, absolutely. And so, yeah, when it comes to metabolism, right, you make the phenol. And this is the main issue. It's not really the methoxy group per se, but it's the phenol portion. And so you really need to stop that first demethylation somehow. And so hopefully these strategies help address the O demethylation problem. There are phenols in drugs. It's not like there's no phenols in drugs, but
Yeah.
yes, making a quinone type species is quite the, you have to worry about that as a toxicity or liability.
Yeah, definitely. Context dependent again. All right. Next question comes from Charles Michael. Um, have the natural products been analyzed to check their content of chloro and methoxy groups? So, like in general, natural products.
Yeah,
You
is
mentioned.
there a review specifically for chloro and methoxy prevalence in natural products? I don't think there's specifically a review. I know, of course, there's people who like to work on, chlorine-containing natural products. Chris Vanderwall comes to mind a lot. Um, he made a lot of chlorosulfolipids, for example. If you speak to Dennis Hu, I'm sure he'll talk about how Noah Burns was looking at chlorinated molecules as well. Yeah, I think Eric Carrera did some of that too. And so, yes, there's definitely instances of it. And you might have reviews of certain classes of natural products, but maybe not a general review on natural products with chlorine or methoxy.
Sounds good. Yeah, I haven't seen one of my own either. So yeah, it seems to be pretty uncommon. Yeah, we have many more questions, maybe a few more.
Yeah.
You already touched on the bromine, right? So bromine atoms and drugs. Can you maybe talk a little bit about in depth or what you know about why they are often seen less?
Yeah. So I don't think bromine is inherently bad. Of course, you don't want it on alkyl system. I definitely stay away from alkyl bromides, first of all, since that's just chemical reactivity. On aromatic rings, right? It's not like a bromine would necessarily pop off in an SNAR reaction, or, of course, you're not going to put palladium in your body. So I think that's okay. But it's just the mass as well, right? And so I wouldn't put, if you were to put, I wouldn't put more than one bromine on a molecule, but it can have magic chloro-like effects. Sometimes the matched molecular pairs have bromine showing similar potency improvements.
Perfect. And another question about the metabolism. Can you improve metabolic stability by increasing the steric bulk around the methoxy? Does it also work to increase steric bulk around the ether group itself, e.g., an isopropyl instead of a methyl if potency remains similar?
Um, the isopropyl group typically makes things worse, actually, because it's about the bond association energy of the CH bond in a radical fashion. And so, of course, if you have a tert-butyl ether, then you kind of block that CH bond, right? But an isopropyl ether typically makes it worse. Even if it's sterically hindered, it's really that BDE energy that you're looking at.
Great. Yeah, we are actually at time. So we have a few more questions. I think Yoshi would be very happy to answer questions probably offline, and we'll probably make a collection of the questions and write some answers on Drug Hunter. So
That would be good. Yeah, if you can actually collate them from me and then I could answer them, maybe put them in the talk feedback or something along the lines, I'd be happy to do that. Or people can reach out to me on LinkedIn.
awesome. Thank you so much. Well, I want to thank you, Yoshi, for a phenomenal talk. I really learned so much and really very nicely illustrated those molecular interactions, the physicochemical properties, and the metabolic properties and everything really. And you mentioned in the beginning of the talk that this may not lead to more drugs that contain chloro and methoxy groups, but I do think that the audience will be inspired to do more designs of chloro and methoxy groups. So thank you a lot for a phenomenal talk. Thanks everyone in the audience for dialing in. And yeah, I wish everybody a wonderful rest of your week, and please dial in to our next event.